Hello, everyone. Welcome to today's live broadcast, Unraveling the Structure of a Monoclonal Antibody, a targeted bottom-up and top-down analysis of trastuzumab and glycosylation variants. I'm Steve Brown, Technical Editor of LCGC, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LCGC and sponsored by MacMod. A company should be able to condense its mission statement into a simple phrase. MacMod's mission statement is smarter chromatography. But just what does smarter chromatography mean? It means that when they make a recommendation to you about an HPLC or UHPLC product, it is to partner with you to offer innovative solutions to your complex problems. They use advanced state-of-the-art analytical software to match their product portfolio with your separation needs. They leverage their 29 years of experience in the LC column business, their technical expertise, and manufacturing network to help you solve your toughest application problems. MacMod provides up-to-date and accurate technical catalogs and reports from industry-leading separation scientists to keep you informed of new technologies. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found on the right-hand side of your screen. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the small green icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark help widget in the dock at the bottom of your presentation window. Please note the valuable resources available on the left-hand side of your screen provided by our sponsor. I would now like to introduce today's speaker. We are pleased to be joined today by Barry Boyes. Barry is the Director of R&D for Biosciences at Advanced Materials Technologies in Wilmington, Delaware, Adjunct Professor of Chemistry at the University of Georgia, and the CSO of GlycoScientific, LLC. Barry started his career in biochemistry at the University of Alberta, completing his PhD in neuroscience at the University of British Columbia under the supervision of Professor Edith McGeer. After work at Central Research at the DuPont Company in Wilmington, Delaware, he joined Drs. Kirkland and DiStefano at Rockland Technologies, where he developed a successful program to commercialize a variety of separations products and technologies for proteins and peptides. HP acquired Rockland in 1997, and Barry stayed with the company, which later became Agilent Technologies. At Agilent, Barry served a variety of roles in R&D and product development, becoming the Senior R&D Manager for Separations, Consumables, and Services, then the bioreagents business, bringing to market a range of nucleic acid and proteomics analysis products. In 2006, he became the VP of R&D at the Edgewood, Maryland site of Smith's Detection, commercializing field-ready biothreat detectors for infectious and toxic agents using a variety of biochemical and spectroscopic technologies. In 2009, Barry returned to full-time pursuit of technologies for high-resolution biomolecule analysis using novel technologies pioneered at AMT for superficially porous particles. Since then, his efforts have focused on peptide, protein, and glycoprotein analysis using LC and LCMS. Barry, thank you for joining us today. Please get us started.
Okay, thank you, Steve. Sorry for that long-winded introduction. Today we're going to be talking about essentially liquid chromatographic and mass spectral methods to study the structure of proteins, and these will be exemplified with some applications regarding monoclonal antibodies. The majority of the work that we'll be discussing concerns the use of superficially porous particles and of the halo products, which for bioanalytical applications are referred to as the halo bioclass fused core products. On the agenda today, we're concerned with dealing with the rather challenging prospects for the detailed analysis of proteins. Proteins can be challenging subjects because they are, of course, high molecular weight materials which are polyelectrolytes. This can affect the peak shape as well as the ability to detect proteins by electrospray ionization, a common method for mass spectroscopic analysis. Proteins have the characteristics of considerable opportunity for heterogeneity due to post-translational modifications as well as chemical modifications, predominantly through oxidative mechanisms. But as well, there can be additional heterogeneity introduced during bioprocessing and other upstream considerations. Because they are proteins, they are subject to change for a variety of environmental reasons, including storage, including processing, and including means of bringing them into solution or formulating biotherapeutic proteins. So often, we're required to use a combination of methods and approaches to sort out this high degree of potential complexity. Now, in our favor, there have been a number of recent enablers that allow detailed protein structure analysis. Of course, the most attention in the last decade has been around the very considerable improvements in mass spectroscopy analysis. And it's fair to say that I would expect for this to continue. There has also been significant improvements in liquid chromatography stationary phase development. So LC has improved as well, both in terms of the materials for conducting the separation, that is to say stationary phases, as well as the instrumentation itself. What has been a bit lagging has been development of alternatives in mobile phase characteristics. That is, we've improved the detection, we've improved the stationary phases, we've done relatively little to improve mobile phase or other operational conditions. We'll talk a little bit about that today in terms of some of the recent developments for mobile phase modifications. Our focus at Advanced Materials Technologies has really in the last decade centered on biomolecule applications. And a lot of this work has been concerned with further improving column packing materials and liquid chromatography columns. This is our primary commercial activity. But we have spent quite a bit of time and resource to finding useful conditions or trying to expand the useful conditions for conducting separations for proteins and for peptides, as well as nucleic acids. We have, over time, developed more of a workflow view of protein bioanalytics, and we'll discuss that in some detail towards the later part of this presentation today. Now, as I mentioned, our primary commercial activity at Advanced Materials Technologies has been concerned with the improvement of liquid chromatographic stationary phase materials, that is, column packing materials, and the means of putting them into columns for useful application. A lot of this work was driven off initial rapid success and acceptance of high-performance superficially porous particulate column packing materials, which in our case are known by the trade name HALO fused core materials. There are a number of other trade names that have entered the commercial marketplace in 8 years since we initially commercially introduced such materials. Now, the defining characteristic of the superficially porous particle is the solid core of silica, shown in cartoon form on the left-hand side of your screen, surrounded by a superficially porous shell of silica microparticulate structure. This is the porous material that allows molecules to diffuse in and out of the superficially porous particle and to interact with a stationary phase that has chemically bonded to those microparticulate shell structures. On the right-hand side, you'll see that this is not merely a cartoon representation, but does reflect the way that the materials are actually built and used in liquid chromatography. That being a scanning electron micrograph of a particle that's been cut in cross-section. And you can see the dense core in the center of the particle surrounded by the porous, superficially porous shell. Now, we liked these materials and they have, as I mentioned, been rapidly accepted for small molecule liquid chromatography over the last decade because relative to much smaller totally porous particles, they exhibit a relatively modest back pressure drop across the column due to the particle design. And a way of looking at it is that the column efficiency of a superficially porous particle in actual use is equivalent to a much smaller totally porous particle. The disadvantage of such so-called sub-2 micron or micro particles being that they do generate significant back pressure. And during the time in which so-called ultra-performance or ultra-high performance liquid chromatography was taking off in the marketplace, when instrumentation was relatively less common than it has become in the last couple of years, the superficially porous particles delivered the equivalent column efficiency and performance to the sub-2 micron particulate materials that were required for improving performance. Because these are relatively larger particles compared to sub-2 micron particles, there are a number of practical advantages, including the fact that the bed itself is more permeable. That, of course, improves the column back pressure, but it also means that there is less demand in terms of eliminating small particulate contaminants that could plug the column. The implication of this particle morphology is also that as you go to increasingly high linear velocity, that is flow rate, high resolution is maintained, and the performance advantage is maintained as analysis speed is increased. Now, as I mentioned, much of the last decade has been spent on really looking at the question of, well, we had an unanticipated improvement for small molecule applications with such superficially porous particles. Can those advantages be translated to the analysis of larger biomolecules? Or, as I say in this slide, can superficially porous particles fit the needs for protein science? Now, I'm originally a protein biochemist, so the opportunity to improve resolution or to improve performance would be a welcome one for anybody who studies complex protein preparations. So if we're to apply such materials, what are the needs in terms of the engineering of the materials to go into a chromatography column? Well, the first is we're now talking about larger molecules, so the pore size must fit molecular size, that is the hydrated volume that will be occupied by larger proteins or nucleic acids or peptides. If we do not have this, then restricted diffusion will limit the column efficiency and the loading capacity of the chromatographic material. The net effect of that being that peak capacity, the ability to resolve peaks, is going to be limited by kinetic and retention problems. Particle geometry, in order to meet the requirements for analytical purposes, must optimize the surface area to volume characteristics of the particle. That is, we have the shell thickness to consider. Thickness and the pore size will determine the diffusion limits for larger molecules, whereas the surface area will have a direct impact on how much material we can load on a particle before we begin to overwhelm its available surface area. That is the interactive sites. Part of the equation for optimizing such materials is the core size. That is, if we increase the core size too greatly, we decrease the shell thickness, and we increase the overall particle size, leading to a loss of chromatographic performance. We must balance these various characteristics of the particle in order to obtain the most useful material, and we must do so in a way which allows it to be manufacturable, that is, to have a desirable particle size distribution and minimize the variance in particle characteristics between particles. Now, having done so, of course, we must have surface chemistry that is appropriate to the sample. That is, what has the desirable characteristics to interact with proteins or other bio. Now, we demonstrated this relatively early on. In this case, the data that I'm showing you is published in 2012. There's a reference to the bottom right on the slide where we looked systematically at pore size as well as shell thickness and other characteristics on the efficiency of separations as we varied the size of the molecules that we wanted to resolve. In this case, comparing the effects of mobile phase velocity, that is flow rate, on a measure of column efficiency, which in this case is the reduced plate height. Looking at a small peptide, leucine and kephalin, about 600 molecular weight, and comparing it to the beta-amyloid 1 to 38 sequence, which is about a 4,000 molecular weight material. If we look at the effects of column efficiency versus flow rate, we see that a larger pore, which is the middle curve, does much better than a smaller pore, which is the top curve, when applied to this larger analyte, that is beta-amyloid. And the reason for this is that restricted diffusion phenomena that I referred to. And you can see by matching the correct pore size to molecular weight relationship, we get a considerable improvement in column efficiency and therefore the ability to resolve samples. In this case, nearly 200% lower plate height, that is improved efficiency, with the appropriate particle or pore size. Now, in practice, what this means is that as we go to higher flow rates, we can maintain the separation capability of an appropriately designed material. In this case, we have a 2.1 millimeter ID column with a tryptic digest separation conducted under typical conditions for such digest, that is trifluoric acid, and looking at a range of flow rates from rather fast, which is 0.75 milliliters per minute, to fast, to even faster, 1 milliliter per minute, and 1.5 milliliters. So even though we are operating at nearly tenfold above so-called typical conditions, we still maintain considerable ability to resolve a sample. This is often overlooked for people who have used superficially porous materials, that because the dependence on flow rate is more decoupled from column efficiency, that fast separations can be conducted for complex samples. We have applied this logic to look at materials appropriate for larger biomolecules. That is, I showed you an example of tryptic digests or relatively larger peptides, about 40 amino acid residues, but what happens when we need a material appropriate for intact proteins or subunits of protein, ranging anywhere from 10 to 100,000 molecules or larger. Well, in this case, not surprisingly, we have to further increase the pore size of the superficially porous shell. And the size that we have settled on commercially is the 400 angstrom pore size. We came to this by systematic analysis, varying the pore size, the shell thickness, and the diameter of the solid core. And what we have found that a good compromise of back pressure, performance, applicability to protein separations is the particle that's shown diagrammatically on the left side of the screen. That is 3 micron core, a 0.2 micron porous shell with 400 angstrom pore sizes for a net particle diameter of 3.4 micron. Micrographs on the right show that these have been reduced to practice and that they are described by our design criteria. So again, because these are larger pores than sub 2 micron materials, We have a low back pressure, good calm permeability. We do have considerable forgiveness with respect to particulate contaminants that may be in the flow stream and entering the HP column. Our design in this case identified that the shell thickness in particular, as well as the pore size, was critical to obtain a material that shows the highest performance. And we see a comparison in this case at rather high flow rates, in this case 1.1 mils per minute with a 2.1 millimeter ID column diameter, so that's a pretty high flow rate, using a gradient separation and separating a mixture of widely commercially available proteins. The numbers over top the chromatograms showing the comparison of a 3.4 micron, 400 angstrom superficially porous material to that of the 1.7 micron, 300 angstrom totally porous are the peak widths. So a measure of column efficiency, column performance. And as you can see that at the rather high flow rate, in all cases, protein bandwidth, that is the broadness of the peak, is reduced for a superficially porous material. Moreover, the back pressure, as measured at the LC pump, is considerably lower for the 3.4 micron particle diameter. So there is an advantage, even relative to much smaller particles, for a superficially porous structure. Here's an example showing the resolution of main peak and some surrounding contaminants or variants for a monoclonal antibody preparation, in this case the Siluumab material, which is commercially available from Sigma, the humanized monoclonal antibody. And you can see on the top panel the separation conducted 3.4 micron superficially porous material we refer to as the protein C4 to a sub 2 micron C4 material with 300 angstrom bottom panel. Significant improvement in terms of peak characteristic separation properties. Now, one of the questions that I'm often asked is, well, is improving protein separation characteristics more dependent on particle size, pore size, or in the case of superficially porous materials, on the relationship of those various parameters? And the answer is, well, they all have contributions to make. So in this case, I'm showing the separation conducted with a 2 micron superficially porous material with 400 angstroms and calculating the peak capacity, which is essentially the average of all of those proteins contributing to the peak capacity calculation. And we can see that for a 2 micron particle compared to a sub 2 micron totally porous particle, we obtain nearly double the peak capacity. Now, I should include the provision here that this 2 micron material, which is currently in development in our research and development department, should be released early in 2016. So you can see that just decreasing the particle diameter does not optimize protein separations. It requires consideration of the diffusional limitations because proteins are large. Diffusion dominates the ability to resolve such large biomolecules. The diffusion limitations are decoupled to some extent by the use of a superficially porous particle morphology. So we've talked to this point predominantly about characteristics of stationary phases. What I'd like to do is change a little bit here to discuss mobile phase characteristics and specifically focusing on what would we need to do in terms of modifying mobile phase characteristics that might allow us to take even further advantage of these improvements in stationary phase materials, and particularly when keeping an eye on detection using mass spectrometry. So if we look at applications, particularly in reverse phase LC separations, trifluoroacetic acid as a mobile phase modifier greatly dominates analytical applications. And the reason for this is that it does show good peak shape and high column efficiency. Formic acid, to a lesser extent acetic acid, by comparison has been the most widely adopted for LC-MS applications. As you can get reasonable LC performance, and formic acid and acetic acid, small organic acids, are highly compatible with mass spectroscopic detection. Whereas TFA shows considerable loss of signal in LC-MS applications, predominantly driven by the suppression in electrospray ionization due to the rather strong ion pairs that are formed between trifluoroacetic acid and the basic aminoacyl residue side chains on protein or peptide analytes. So that doesn't mean that TFA cannot be used, but in all cases there will be a decreased sensitivity relative to the use of formic acid. So currently, the vast majority of applications of LC-MS use either formic acid or trifluoroacetic acid. And one of the areas that we've explored in detail has been to look at the question of, are there other alternatives? Almost all the examples that I'll show you do not include details on other work that we have conducted on varying the organic modifier, that is the strong mobile phase component, although there are some examples included in the slides that we'll get to, and almost everything that I show you takes advantage of the use of column heaters, that is elevated temperature, for the separation of proteins and peptides, which is just generically a good idea. Our first foray into this really concerned trying to explore the idea that modifying ionic strength of mobile phase modifiers may have utility. And the thinking here was that, well, in many cases, well, in all cases, all proteins and certainly many peptides have basic amino acid side chain groups, and that operation at low pH could have considerable disadvantage with respect to interaction with sites on the stationary phase material, but also at reasonable loading conditions. High loads of such analytes could cause ion-ion repulsion, leading to a loss of chromatographic efficiency. Now, this logic was really an extension, and our experiments in this area were really an extension of work that was initiated by David Macaulay. There's a reference on the bottom of the slide. Hopefully you can see that if you want to look up more detail on this. When he noted that when he was looking at basic peptide separations, that the addition of ammonium formate to the mobile phase containing formic acid, net effect is adding a neutral salt to an acidic mobile phase, greatly improved the peak shape. Another factor that we note on this slide, comparing the chromatograms on the left-hand side of the slide, the middle chromatogram to the bottom chromatogram, is that the addition of that neutral salt, that is ammonium formate, also increases retention, so relative to formic acid. So we get both an increase in retention as well as considerable improvement in the peak shape, that is, the asymmetry of the peak and the sharpness of the peak. So this worked out fairly well. We've described it in a publication showing at the bottom of the slide. And we also took the analysis a little bit further. By the way, the reason why we thought that this would be an okay idea was that we knew from many years of work on small molecule LC-MS applications that ammonium formate is not terribly problematic with respect to online mass spectral detection. So we did all of this work with online, in this case with an ion trap as the mass spectral detector, and detailed not only the characteristics with respect to separation properties, but also the mass spectral detection properties. As you can see, comparing formic acid on the left side of the screen to mixture of formic acid and ammonium formate on the right side of the screen, that there is limited effect detectability in mass spectroscopy, but also the ionization state characteristics in MS or in MS-MS mode are not negatively impacted by the use of ammonium formate acid phase. So essentially this simple manipulation improves chromatographic performance even visually. The question became, and a driver for this was, well, since we're now improving peptide separations, will it have a net benefit on proteomic analysis? That is, the separation of a complex mixture of peptides. And we have detailed this in Journal of Biomolecular Techniques, where we looked at a tryptic digest separation, complex mixture that results from the tryptic digest of standard proteins, as well as an actual proteomic sample, in this case from canine prostates, looking at the quality of peptide mass spectral identifications and the resulting protein IDs that come from that analysis. And to summarize a series of publications in this area, the net effect is that the improvement in chromatography leads to significant improvement in proteomic analysis. So with those improvements now in hand, with a relatively straightforward adjustment of something like ionic strength, it has caused us to rethink whether or not there may potentially be additional improvements from mobile phase manipulations that could be applied to protein analysis. And if we were to do this, we must restrict ourselves to materials that are compatible with mass spectroscopic detection. Well, the first property that should be considered is, of course, volatility. We are doing electrospray ionization. Anything that we spray into the electrospray interface is going to end up in our mass spectrometer ion path. If it is not volatile, we will cause considerable problems with online detection. And, of course, keeping our mass spectrometer clean. As well, there's the consideration of, under what conditions do we get good ionization? Well, generally low pH. There is the neutral or elevated pH will give reasonable ionization for many, but not all proteins or peptides. And the collective experience is that certainly for reverse phase separations, that there will be a considerable problem in peak shape and chromatographic efficiency. The biggest consideration, though, is if we're to use new mobile phase modifiers for protein separation, we must really consider solubility. We have materials that we are injecting onto an LC system that are not fully soluble. We will certainly have, at very minimum, complex peak shapes and recovery problems. And, in fact, this could extend to just blocking the flow path and overpressurizing the system. So as we begin to look at different alternatives, we have that additional set of criteria to consider. Now, we've looked at a lot of different materials for doing this, and I will summarize by showing you some of the observations that we have been able to make in this area with a restricted set of additives that I'll go into more detail on. So in this case, we are comparing formic acid, acetic acid, trifluoroacetic acid, as well as two compounds that you may be less familiar with, difluoroacetic acid, shown fourth from the left, and first on the right, 3-fluoropropionic acid. All of these candidates have the desired characteristics of being volatile, relatively lower pKa. The pKa's are shown under the abbreviation for the compounds, and all of these will support quite reasonable protein solubility, with the exception of acetic acid, which we have found to be variable. Okay, I'm going to summarize some results here graphically. On the left-hand side, we're showing the results obtained with UV detection at 280 nanometers, comparing these different mobile phase modifiers. And on the right-hand panel, we're showing the total ion current generated in mass spectrometer, which is operated in line with the UV detector. So the left-hand side gives us an impression of generic recovery. The right-hand side gives us an idea of the ability to generate a useful electrospray ionization mass spectral signal. And as you can see, looking at the left-hand side, some of these mobile phase modifiers are better than others with respect to peak shape and column efficiency. Trifluoroacetic acid is always the best that we've found in terms of giving good peak shape and retention. Formic acid, not nearly as good. Difluoroacetic acid, more like trifluoroacetic acid than formic acid, and 3-FPA, more like formic acid. In terms of the mass spectral data shown on the right-hand side, trifluoroacetic acid is very poor, formic acid is the best, and we see intermediate results using difluoroacetic acid or fluoropropionic acid. Do these different additives have any effects on the ionization state? This is a quick comparative analysis showing the average state. That's the numbers that you'll see towards the right hand of the MS data for these eluted protein peaks. The answer is very subtle effects with respect to the ionization envelope, and there's certainly no great effects. The largest being that comparing trifluoroacetic acid to formic acid, where we have a shift to lower number of charges per protein molecule. But the overall effect on ionization is subtle. What about overall ability to detect in mass spectrometry and overall effects in terms of chromatographic performance? And I've summarized that in these two bar charts. On the right-hand side, looking at the peak width, that is, smaller is better, comparing these acid modifiers to formic acid, which is 100%. So if we look at trifluoroacetic acid or very similar difluoroacetic acid, we get about a 2 to 3-fold generic decrease in peak width. So that's quite good. In terms of the cost for mass spectral detection characteristics, for difluoroacetic acid, we have about a 5 to 10-fold decrease. With trifluoroacetic acid, more like 20 to 40-fold decrease in terms of detection sensitivity. So the net effect there is that difluoroacetic acid causes some ionization suppression, not nearly as much as trifluoroacetic acid, but we can still use that for mass spectral detection. Now, Why would we want to do that? Well, here's an example showing separation of hemoglobin subunits, comparing formic acid under desirable formic acid conditions, that is about 50 millimolar, or 0.2%, and you can see fairly broad peaks compared to 10 millimolar difluoroacetic acid, shown in the middle panel, or a mixture of difluoroacetic acid and formic acid in the bottom panel. The summary here is that all of the, and by the way, on the left-hand side we have the intensity for the mass spectral detection. And you'll note they're all on the same scale, first of all. So even though there's significant ionization suppression by difluoroacetic acid, there's still enough detection sensitivity, and the marked improvement in peak width is enough to make detection characteristics similar to the intensities that we see with formic acid. But most importantly, we're beginning to pull variants of the different subunits of hemoglobin away from the main peaks, where we can realistically obtain mass spectral data so that we can assign or assess the correct molecular mass for those protein variants. So there does seem to be advantage to the use of alternate mobile phase modifiers for intact protein or protein subunit separations. Now I'm going to, again, switch tracks here a little bit to look specifically at the workflows for which we can apply both these improved chromatographic materials as well as mobile phase modifiers for monoclonal antibody analysis. And there's essentially three application areas that we'll be talking about. That is the use of the application of very wide pore materials for intact protein and subunit separations. That is to look at impurities and post-translational modifications using the halo protein, to identify impurities and modifications using the halo peptide, and to look at glycosylation variants using the haloglycan column. So this slide shows a rather straightforward separation of trastuzumab tryptic digest. In this case, the intact monoclonal antibody is digested with trypsin and then subjected to separation on halopeptide column using a formic acid ammonium formate containing mobile phase. This allows mass spectral coupling as well as the use of UV detection. And you can see that there's excellent resolution at reasonable separation conditions, in this case less than an hour resolution time, and very considerable peak capacity to separate the cryptic fragments. We can also couple to high-resolution mass spectrometry such separations. In this case, the same kind of a digest, that is of trastuzumab tryptic digest, but in this case using a very long column of 5-micron particle size halopeptide ESC18. We've put together two 250 millimeter columns for 500 millimeter, 50 centimeter total column length, and this is upstream from the Thermo Velos Pro Orbitrap mass spectrometer. So we're able to fragment the resolved peptides as well. On the bottom panel, you'll see the base peak chromatogram for that separation. So looking at the MS signal, and above it, the mass fragments from a selected peptide where we can now read off the sequence and identify by spectral ID matching. So we use this approach to look at the sequence coverage as well as to identify protein chemical variants from chemical modification variants that may be present in the preparation using tryptic digests. We can also apply some of these improved separation conditions and materials to look at the subunit composition of monoclonal antibodies. In this case, we have a reduced monoclonal antibody where we're separating the light chain and heavy chain subunits, and in this case, using straightforward trifluoroacetic acid conditions with the haloprotein C4, we're able to resolve heavy chain variants because we're using trifluoroacetic acid. Not nearly so good in terms of mass spectral application, but certainly we can now see that there are variants present. If we want to use mass spectral detection, we need to use some of these considerations of other mobile phases than trifluoroacetic acid. In this case, this is again an example that's been published with a reference on the bottom right. We're using a formic acid modified mobile phase, in this case a fairly high concentration of formic acid, with a flatter gradient condition to resolve the light chain and some of its variants, and a heavy chain and some of its variants, truncated sequences and other modifications. So we've looked at that kind of separation in a little bit more detail with respect to are there better or worse conditions that we could apply for mobile phases using the superficially porous protein C4 column. In this case, we're looking at reduced Silumab, again, that commercially available standard from Sigma Biosciences, and resolved using a formic acid ammonium formate modified mobile phase on the top, so similar to what we've just seen, difluoroacetic acid in the middle, and formic acid on the bottom. And consistent with our previous observations, formic acid is relatively poor in terms of its chromatographic performance. Difluoroacetic acid shows some potential advantages, and formic acid modified with ammonium formate does improve the separation. Because we're now looking at a mobile phase modifier in difluoroacetic acid that is more compatible with mass spectrometry, we can couple that separation condition, in this case with the Orbitrap, and start to deconvolute the mass spectral data in order to obtain some information on the modifications, in this case glycan modifications, that we can pull out of the heavy chain mass spectral data. Now, we've applied the same logic to looking at intact monoclonal antibody separation. In other words, manipulating mobile phases to see if we can improve intact protein separation. In this case, again with Silumab, operated using trifluoroacetic acid, difluoroacetic acid, or a mixture of difluoroacetic acid and ammonium formate and formic acid. So kind of all of the above. And you can see that difluoroacetic acid by itself shows some advantages, we know, in terms of mass spectral detection capabilities, and similar characteristics to the excellent performance that we get with trifluoroacetic acid. As we've tried to modify that further, in this particular example, it has not been efficacious by adjusting the ionic strength of the ammonium formate. Now, we need to be careful when we look at intact protein separations, because in addition to the variants that we know are present in intact proteins, that is, potential N-linked glycosylation variants and chemical modification variants, or maybe even sequence modification variants, if we manipulate the operational conditions without some caution, we could run into what I believe to be the problem looking at this comparison of conditions. In this case, looking at trastuzumab with a fairly steep gradient, a flatter gradient, and then on the bottom panel, an even flatter gradient. So in this case, we're just varying the rate at which acetonitrile is varied. In other words, the gradient steepness. In the bottom panel, we're beginning to see a lot of what look to be potentially variance come away from the main peak. The problem is very likely that we're beginning to induce artifactual separation of folding intermediates or conformational variants. And we must remain cognizant of this as we begin to manipulate mobile phase and operating conditions. At this point, we do not know what all of those variants are that we see in this TFA, modified mobile phase separation, are, but it does identify that we should be concerned to not resolve things that potentially are not variants that we understand. One of the systems that we've looked at in considerably more detail now is using difluoroacetic acid. And in this case, I'm showing this example to identify that even though we have designed materials with large pore sizes and thin shells and improving the kinetic conditions that will allow higher resolution, flow rate still remains a considerable variable in terms of being able to get maximum resolution out of any chromatographic material when you have an analyte, which in this case is 150,000 molar weight. So on the left-hand side, we're showing gradient, which is operated at 0.4 mils per minute and run in 20 minutes to same resolution or same gradient conditions in 40 minutes at half the flow rate. So the net effect is the gradients are comparable, but delivered at two different flow rates. And you can see that there's significant improvement in resolution on the right-hand side. And this is because even though we have gone to great lengths to improve the diffusion situation, that is the kinetic limitations of the separation, there are still limitations with respect to how fast you can use a molecule into and out of even a superficially porous structure. But we are beginning to resolve some interesting species from that trastuzumab preparation. And this does allow us to then apply high-resolution mass spectroscopy online to define what exactly are those things that we're beginning to resolve. And that is an ongoing research question in our lab. I'll show you how we're attacking it. In this case, on the right-hand top panel, we see a typical high-resolution mass spectral data, in this case collected in difluoroacetic acid-containing mobile phase. We've blown up the mass spectral data under that panel so that we can see the charge state. We've then deconvoluted it using protein deconvolution version 4 from Thermo, and we're able to resolve the N-linked glycan variants on the antibody. I'd like to direct your attention to the blown up mass spectral data where we see that there where we see what looks like a mixture of signal and noise. And the reason why I'm drawing your attention to that is not to say that I can't collect decent quality mass spectral data, but to point out that there may be not just noise in there, but complexity that results from the complexity of the analytes that we're undertaking LCMS with. And what I mean by that is these are monoclonal antibodies, and these monoclonal antibodies are a mixture of N-linked glycosylation modified proteins. And as we usually look at monoclonal antibodies when we do glycan analysis, we typically see 2 or 3 main peaks in terms of the end-linked glycans. In the case of trastuzumab, we typically will see 5 peaks here. The G0F, a mixture of 2 different isomers of G1F and the G2F. And these will lead to differences in net mass of the monoclonal antibody. And there will be a mixture of these glycans when we look at the intact monoclonal antibody. What I'd like to point out is that now we're blowing up the data, we're looking a little bit closer to it, is that in any of these preparations, it's not just those 3 different masses of glycans, there's a number of less common variants at the glycosylation site. And we know what these are in this case because we've collected mass spectral data in series with the fluorescence data for these end-linked PNGase F-released glycans. So we must recall that all of these additional glycan species are contributing their mass differences to that high-resolution mass spectroscopic data. So the task of looking at an intact monoclonal antibody is not simply deconvoluting, but there is a limitation implied by not only the glycan heterogeneity, but considerable potential polypeptide heterogeneity at each of those glycan variants. So what I'm trying to convince you of is that what we see as potentially noise in high-resolution mass spectral data is really more a limitation that centers on the complexity of our samples than it is the lack of performance high-resolution mass spectral analysis coupled with high-resolution LC separations. So let me summarize. With respect to observations for monoclonal antibodies and the subunit and cryptic digest analysis, it's clear that trifluoroacetic acid is not required for high-resolution separations. I've shown examples of high-resolution separations using modifiers other than trifluoroacetic acid, one of which is difluoroacetic acid. Electrospray ionization is possible and actually quite reasonable with difluoroacetic acid. In terms of how we deal with high-resolution data from LCMS, there are questions that come up unless we're very cautious about what exactly are the species we're separating, and is generating more peaks always better? And the answer is not unless we know what those structures are. And that because of the complexity of our samples, we must approach deconvolution of high-resolution mass spectral data very cautiously. And overall, to conclude, some of the bottlenecks in protein LCMS have been improved with newer superficially porous materials, and we've shown examples of that. There are a wider range of other operational conditions that you can take advantage of and combine with these higher resolution superficially porous materials to overall increase protein separation capabilities. In some cases, we're now getting sufficient resolution that we should proceed with caution to be very clear on what it is that we are resolving and what it means, because it's not always the case that an additional peak represents a unique and known protein structure. To resolve all of those structures, we will need to undertake considerable greater analysis. And I'd like to thank my various collaborators as well as the NIH for their financial support. And with that, I will conclude and turn the presentation back to Steve.
Thank you, Barry. Before we get started with the Q&A portion of the presentation, we're going to ask the audience 2 brief polling questions. You can click directly on your screen to submit your answer. First question, which product today did you find most interesting? Halo protein 400 angstrom C4 and ESC18 3.4 micrometer bioclass fused core column, Halo peptide 160 angstrom ESC18 and ESCN 2.7 and 5 micrometer bioclass fused core column, or Halo glycan 90 angstrom 2.7 micrometer bioclass fused core column. Second question. Which topic would you like to learn more about? Novel mobile phase additives to improve mass spectral analysis, top-down approach to analyzing monoclonal antibodies by reverse phase LCMS and LCUV, bottom-up approach to analyzing monoclonal antibodies by reverse phase LCMS and LCUV, analysis of glycosylation variants of monoclonal antibodies, or selecting the right Halo bioclass fused core column for your bioseparation. Thank you for taking the polling questions. At this point, I would like to remind the audience about how to submit questions. You can submit questions by typing them in the Q&A box, which can be found on the right-hand side of your presentation window. We'll try to answer as many questions as we can. Our first question for Dr. Boyes. Well, let's see. What challenges did you run into when using columns with 0.2 millimeter internal diameter? Was the HPLC system suitable in terms of minimum dispersion?
Okay, well, let me tackle that by saying, whenever going to capillary columns, yes, you need to take considerable efforts in considering band dispersion, particularly on entrance to the mass spectrometer. Now, we used a trap pre-column strategy, so we were able to eliminate most of the pre-column band dispersion by essentially plumbing it through a trap column in front of the analytical column. The post-column, we've mostly operated in two modes, one with a UV detector in series and one where we plumb directly to the mass spectrometer. Now, what we find is that even with the smallest volume UV detectors, there's a significant increase in band dispersion before the mass spec. So we, in order to obtain the best data, go directly into the mass spectrometer. Now, with capillary columns, it really is more an issue of how exactly you've put together your hardware. There's a variety of different commercially available interfaces. In our case, we use one that's relatively off the shelf and have minimized the band dispersion for, but it's always going to be a problem when you're interfacing mass spec.
All right.
So we're actually redoing some of the experiments that we have completed with capillary columns with larger internal diameter columns, so we can get even better data from some of those closely eluting peaks, essentially by just really taking down as much post-column dispersion as possible. Don't know if that answers your question. Are there challenges? There absolutely always is when interfacing mass spec.
All right, great. Thank you, Barry. Next question. Are the new mobile phases compatible with detergents like Lubrol or Chaps?
I do not know because I haven't used those detergents particularly in my mass spectrometer because I tend to shy away from putting detergents anywhere near my mass specs. But I wouldn't imagine that they would show any remarkable differences compared to trifluoroacetic acid.
All right. Next question. You described the use of some unique acids in the presentation. Where can these be obtained?
Oh, okay. Well, they're actually commercially available through Sigma Scientific. as well as a company called Fluorosyn, which is located in Florida, and I think you can pull that actually off a Google search. But I know that Sigma has them as well. We've compared them, and they look highly comparable in terms of their quality and cost.
All right. Thank you, Barry. Unfortunately, we're out of time for more questions. We'll try to follow up offline with any additional questions coming from the audience. I want to thank the audience for their interesting questions and for participating in today's event. I would also like to thank our sponsor, MacMod, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through October of next year. You will receive an email from LCGC alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. We'll see you next time. Goodbye.